Now we'll see about the short answers, that is the 4 marks answers in the botany second year. As per the exam point of view, important 4 marks we are seeing now. In that phenomena, in the first unit, there is a plant physiology. Transport in plants and mineral nutrition lessons are not there in our syllabus. If you are seeing the third lesson, there is the enzymes. In the enzymes particularly, about the different types of the inhibitors of the enzymes. Competitive inhibitors we had, then uncompetitive inhibitors we had, or non-competitive inhibitors, and then feedback inhibition. 3 types of the enzyme inhibitors. In that phenomena, competitive inhibitors are the inhibitors of the enzymes. They are what they are actually. In that phenomena, what is happening here, whatever the enzyme inhibitor which we had, it is structurally similar to the substrate, or it is structurally similar to the enzyme particularly. If you are taking the enzyme, if you are taking the example of the competitive inhibitor, what is happening? The competitive inhibitor is structurally similar to the enzyme we are saying. If you are taking the example, malonate is the competitive inhibitor for the enzyme succinic dehydrogenase. That means it is not allowing the substrate to bind to the active site now, right? Then non-competitive enzyme inhibition, it always includes what? Whatever the enzyme inhibitor which we had particularly, it is binding to the other than active site. That means it is not allowing the substrate to bind to the active site here. Examples of the non-competitive inhibitors, if you are taking metal ions, we can take the examples of the non-competitive inhibitors. Then feedback inhibition. The feedback inhibition, always it includes what actually it means, the end product of the particular reaction. This end product is not allowing the first product to express. If I are taking the example particularly, the glucose 6-phosphate, the accumulation of the glucose 6-phosphate particularly, it is not allowing the hexokinase enzyme to deliver its action. That is the feedback inhibition we are saying particularly. It is inhibiting the glucose 6-phosphate accumulation, is inhibiting the action of the particular hexokinase enzyme. These are the 3 types of the enzyme inhibitors. That is the 4 marks important. And then about the coenzyme, cofactors, all these things we had, that are also important in this thing. And enzyme action, mechanism of enzyme action. In the mechanism of enzyme action, mainly particularly what is happening, it means enzyme plus substrate gives rise to enzyme substrate complex. Enzyme substrate complex gives rise to enzyme product. Enzyme product lastly will be separated. There is a mechanism of enzyme of action. In the mechanism of enzyme action, how the enzyme is combining with the substrate, and how the enzyme substrate complex is separating, how lastly the product is separating, that we have to write in this enzyme mechanism of enzyme action. In the same mechanism of enzyme action particularly, we have to very important thing, that is a graph which we are drawing between the potential energy and then the enzyme action particularly. Activation energy is very important here. That means enzyme action, in the presence of activation energy, for the conversion of the products, it takes less time. In the absence of the activation energy, the enzyme to convert into the particular product, the enzyme taking the conversion of the reactants into product, it takes a lot of time. That means activation energy is showing the lot of difference on the enzyme action. That we have to write particularly in the mechanism of enzyme of the action. Then classification of the enzymes, sometimes they will ask or sometimes they will not ask here. Classification of enzymes mainly consisting of different things: oxidative lyases, ligases, hydrolases. All these things are the classification of enzymes. So for everything we have to give one example, a reaction we have to write here. This is about the enzymes. After enzymes, if you are taking photosynthesis, we have respiration, we have 2 combining, we are seeing the short answers here. Particularly very important answer in these 2 concepts here actually. Differences between the C4 and C3 plants here. C3 plants, C4 plants difference is very important. The C3 plants particularly, we are saying the C3 plants because the first formed product in the C3 plant stable product is a 3-carbon compound, that is a phosphoglyceric acid. C4 plants are the plants. In this thing, what is happening here, the first formed compound in the C4 plants is the 4-carbon compound, that is oxaloacetic acid. C3 plants are also known as temperate plants, that is they are growing the temperate zones. C4 plants are the tropical plants, and because C3 plants which are there, they require the temperature particularly 10 to 15 degrees Celsius. There particularly 10 to 25 degrees Celsius. There particularly in the C4 plants, the temperature required for them is 30 to 45 degrees Celsius. And in the C3 plants particularly, the primary carbon dioxide acceptor is ribulose 1,5-bisphosphate. There in the C4 plants, the primary carbon dioxide acceptor is phosphoenol pyruvate. And Kranz anatomy is seen in the C4 plants, it is absent in the C3 plants. Photorespiration is seen in what? Is seen in the particularly C3 plants, it is absent in the C4 plants. Then in the C3 plants, photosynthetic production activity is low. In C4 plants, the photosynthetic production activity is more. C3 plants, the cycle always takes place in the mesophyll cells. C4 plants, the cycle always takes place in the bundle of sheath cells. C3 plants is also known as Calvin cycle. C4 plants pathway we are calling as a Hatch and Slack pathway. Mainly here C3 plants examples we can take as a potatoes and all these things, and their C4 plants we can take as a particularly sugarcane, we can take the example C3 and C4 plants here. Apart from that thing, if we can take in the particular photosynthesis and respiration about the amphibolic pathway. Amphibolic pathway is a pathway which always includes what actually? Catabolism, anabolism, synthesis and breakdown of the products. That means carbohydrates, proteins, fats, 3 are synthesized, 3 are actually broken down. Lastly, the Krebs cycle takes place with the help of the amphibolic pathway. Amphibolic pathway is also very important for the 4 marks. Apart from the amphibolic pathway, photorespiration. The photorespiration, remember the point, always takes place in particularly this C3 plants only. In the photorespiration, what is happening here? The oxygen which we had actually, uptake of the oxygen takes place particularly. The Rubisco enzyme which we had particularly, Rubisco enzyme in the C3 pathway, in the C3 cycle particularly, it is meant for the uptake of the carbon dioxide, fixation of the carbon dioxide. Apart from that thing, Rubisco, that means carboxylase enzyme which we had particularly, it is also had a capacity of taking the oxygen. That is explained in the photorespiration here. Apart from that thing, photolysis of the water. Photolysis of the water, it means splitting of the water in the presence of light, we are calling as a photolysis of the water. By the photolysis of the water only, there is a scope of production of the proteins and required ATP and ADH, all these things here. And then about the chemiosmotic theory, and always chemiosmotic theory or Peter Mitchell hypothesis is also important for the 4 marks particularly. The chemiosmotic theory or Peter Mitchell hypothesis which we had particularly, by the proton gradient channel, continuously H plus ions which are produced here, by that thing only what will happen? ATP is produced with the help of the ATP synthase enzyme, that we have to write particularly. And apart from that thing, in this photosynthesis and respiration here, electron transport chain, 4 complexes which we are particularly, we have to explain about them also particularly. And these are important in the photorespiration and photosynthesis phenomena, very important. Then after that thing, the plant growth and hormones, if you are taking that lesson, in that lesson, different hormones we had particularly, auxins, gibberellins, cytokinins, ethylene. Plant growth promoters and plant growth regulators we had, both we had. Remember the point, plant growth promoters particularly if you are taking auxins, gibberellins, cytokinins and then ethylene, both are the plant growth promoters. And abscisic acid is the only enzyme which is a plant growth, only hormone which is a plant growth regulator. And if you see the auxins are the plant growth hormones which are discovered by the FW Wendt. Generally natural auxins if you are taking indolacetic acid, indolbutyric acid we are taking, and artificial auxins if you are taking naphthalactic acid and 2,4-dichloro-diphenol acetic. That thing we are saying as artificial auxins. Auxins always promote the root elongation, shoot elongation, and parthenocarpy. That means in the tomatoes particularly parthenocarpy, without reproduction actually seeds are produced, that is the parthenocarpy we are saying particularly. And then auxins also in turn sometimes they are promoting the apical dominance, that means shoot elongation particularly, and different things has been promoted by the auxins we are saying particularly. And gibberellins if you are taking, gibberellins are actually isolated from the fungi known as Gibberella fugicari. Gibberellins always promotes the internodal length of the in the plants particular, that means they increase the length of the plants when we spray the gibberellins. Gibberellins are also helps in the fruit setting in the grapes and all these things, and bolting technique, that means elongation of the internodal length has been increased by the gibberellins particularly. Then cytokinins if you are taking, cytokinins always promotes the cell elongation, cell division, and cytokinins always, which if you are they will deny the senescence of the leaf, that means aging of the leaf has been delayed by the cytokinins, and cytokinins always inhibits the seed dormancy. And the cytokinins which we are particularly kinetin, in the name of the kinetin we are getting the cytokinins here particularly, they always promotes the plant growth. And then if you are taking the ethylene is actually available in the form of the etaphan. Ethylene always promotes the fruit ripening. Apart from the fruit ripening, ethylene also helps in the plant growth and all these things. This all points we have to cover in the hormones particularly. And apart from the plant growth and hormones, this completes your first unit. And in the second unit, if you are second unit, bacteria and virus is not there. Third unit, if you are going for the genetics particularly, in the genetics, monohybrid cross. Before the monohybrid cross, very important answer, that is actually selection of the pea plant for the hybridization. Pea plant is easily available. Important points of the pea plant. Why the Mendel has been taken as a pea plant for this hybridization experiment we are discussing now? Because pea plant is easily available. Pea plant is actually annual plant. In the pea plant, cross pollination, that means cross pollination can be done easily, and self pollination also be done in the pea plant easily. Pea plant consisting of a bisexual flowers, and the pea plant which we are particularly, it reproduces easily, and in the pea plant life cycle will be short span, it can be produced easily. All these points are considered comfortable for the selection of the Mendel for the hybridization experiment. All these points we have to cover for the pea plant. Then monohybrid cross. Monohybrid cross, it means what actually? 4 marks important. Just we have to draw the Punnett square. We have to cross the T, capital T tall with the capital T small t short dwarf. Then we are getting F1 generation, capital T small t. In the F2 generation, if you are crossing capital T small t with the capital T small t, then what we are getting? Phenotypic ratio 3:1 we are getting. Genotypic ratio 1:2:1 we are getting. Phenotypic ratio 3 tall, 1 dwarf. Genotypic ratio capital T 1, and small t 1, then capital T small t 2 we are getting. That is the monohybrid cross. And codominance, if you are taking particularly, codominance is a phenomena in which what is happening here, both alleles which we are dominant recessive, both are dominating in a same aspect. That is the codominance we are saying. For the codominance example, we have to give particularly example we are taking for the codominance, that is the lentils example, spotted, dotted, CS, and CD. F1 generation CS/CD we are getting, and if you are crossing the F1 generation CS/CD, then same phenotypic ratio, genotypic ratio, both we are getting same in the codominance. That is 1:2:1, 1:2:1. Then incomplete dominance. Incomplete dominance clearly indicates that actually no one dominant or else recessive, no two is equally dominating here. That means incompletely both characters are dominating. New character we are getting in the incomplete dominance. In the Mirabella zelapa plant, we are taking the example. Red colored and white color flowers we are crossing. Red color is indicated by RR, capital R, and white color has been indicated by the small r. When we are crossing with these both things in the F1 generation, we are getting the pink color flowers, that is capital R, small r. In the F2 generation, after F2 generation, the 4 alleles, the 4 traits which we are getting particularly, we are getting the same ratio, 1:2:1. Phenotypic ratio, genotypic ratio also 1:2:1 we are getting. This is about the genetics particularly. And after genetics, if you are seeing the molecular biology, that is the principles of inheritance, particularly in the molecular biology. In the molecular biology, first we are discussing about the Griffith experiment. Sir Frederick Griffith has been proposed that transformation is a phenomena of uptake of the DNA. He has been explained the transformation phenomena by the experiment on the mice. First, what he done actually, Streptococcus pneumonia bacteria he has been injected. S strain, R strain. S strain is the smooth strain, R strain is the rough strain. Smooth strain is the harmful strain, rough strain is the not harmful strain. When the Griffith has been injected the S strain to the mice, then after some time what has been happened? The mice has been died. When the Griffith has been injected the R strain into the mice, what is happening? Nothing has been happened to the mice because R strain is not harmful, we are saying, then mice is alive. In the second step, in the second step, what he done particularly Griffith, it means he took the heat killed S strain and R strain. When he injected the heat killed S strain plus R strain into the mice, after some time he noticed that actually mice has been died. Generally, what should happen? It means if you are injecting heat killed S strain, we are saying that means he inactivated the S strain, smooth strain, and he injected the R strain. When heat killed S strain plus R strain has been injected, in the second step, what has been happened now? Mice has been died, we are saying. Generally, what should happen? Mice should not die because R strain is active there. But what it is proving, what it is showing, that actually transformation has been occurred. That means the smooth strain which has been there, it has been transformed into the R strain. It has been killed the mice. These steps we have to write in the transformation principle. And then about the different thing, if you are taking lac operon concept, we had particularly lac operon concept has been proposed by the Jacobin monad. Lac operon, particularly lac operon, it means lac, it means lactose. Lactose, that means the formation of the lactose, particularly lactose is a disaccharide. It is consisting of glucose plus galactose, two sugars. Lactose operon concept. Operon, it means group of structural genes we are saying. In this lactoperon concept answer, particularly we had IPO in the flowchart, particularly IPO, repressor, promoter, operator. IPO always indicate. Then ZYA we are saying these three are structural genes. Beta galactosidase, transacetylase and permease. Three are the structural genes particularly. In the absence of the inducer, what will happen? It means the repressor which we had particularly, repressor goes and binds to the operator. It will not allow the structural genes to express their genes here in the absence of inducer. In the presence of inducer, what will happen? Inducer will not allow the repressor to bind to the operator, because inducer is binding to the repressor. It is stopping there only. Then what will happen? Structural genes, they will express their enzymes easily. Beta galactosidase, transacetylase, permease. Then what will happen? Lactose, which we had particularly sugar, it is converted into glucose plus galactose. Then glucose will be produced by the lactoperon concept. That is the answer. Then particular differences between the RNA and DNA. RNA and DNA is a common particular answer. That means RNA, DNA. DNA it means deoxyribonucleic acid. RNA it means ribonucleic acid. DNA which we had particularly, it is always present in the nucleus. RNA present in the cytoplasm. DNA which we had particularly, it is double stranded. RNA always single stranded. On the DNA, what is happening here? On the second carbon, OH is absent. Their second carbon OH is present. We are saying particularly DNA is actually stable, RNA is unstable. DNA is not reactive, RNA is reactive. We are saying all these points we have to cover in the DNA and RNA particularly. Then apart from that thing, genetic code. Genetic code is very, very important answer in the particular molecular biology particularly. Genetic code mainly consisting of different postulates. First point is actually genetic code is a triplet code. That means each genetic code is consisting of 3 codons. Totally what is happening? 64 codons we had. In the genetic code, 64 codons we had. And that is actually coding the 20 standard amino acids particularly. In the 64 codons, each genetic code is consisting of 3 codons particularly. And 3 things. Always for the first point of the genetic codon is always universal. All over the world, genetic code is same. Second one, genetic code is actually unambiguous. Unambiguous, it means actually particular codon will be coding for the particular amino acid. Genetic code is degenerate, we are saying. Genetic code is degenerate, it means sometimes one amino acid will be coded by the different codons. If you take the example, glycine will be consisting of 4 codons, phenylalanine will be consisting of 2 codons. And genetic code is always continuous. That means there will be no punctuation marks, there will be no comma, there will be no full stops in the genetic code. Then genetic code always, the initiation codon genetic code is always AUG. And the stop codons of the genetic code is always UAA, UAG and then UGA. See, out of the 64 codons in genetic code, 61 are the sense codons and 3 are the non-sense codons. This is about genetic code. Then if you are moving for the, after molecular biology, if you are moving for the biotechnology, in the biotechnology, if you are taking the 4 marks answers particularly, same the restriction enzymes, about the restriction enzymes, they will give exonuclease, endonuclease. Then about the tools of the RNA technology, that is actually cloning vector, about the structure of the cloning vector they will give. And then the bioreactors, sparging bioreactor and the steroid bioreactor, about the structure. Then they will give about that fermenter bioreactors also, about the 4 marks particularly. And the bioreactor diagram we have to draw, and the blade, sparger blade and sterile blade, that always we have to explain in the bioreactor, all these things. And downstream processing also we have to see particularly in the biotechnology. And micros in human welfare particularly, if you are seeing, then we had about the transgenic plants. Transgenic plants are the plants which are consisting of a desired qualities or desired characters which we require. In the transgenic plants, we have to give the example of the flavor saver tomato, which has a desired character. That is a very, it has a long duration. It can tolerate all the conditions. We can store the flavor saver tomato for the long time. And brassica seeds, which are the highest capacity to produce the new ones. And then Thaipi variety of rice, which is rich in vitamin A, and basmati variety of rice, which can tolerate the biotic and abiotic stress conditions. And different things we have to write about transgenic plants, and all these things. And genetically modified crops and transgenic plants consisting of maximum similar points. And apart from this thing, actually pest resistant crops. The crops which are pest resistant crops, it always includes the RNA silencing mechanism. mRNA silencing mechanism or RNAi mechanism we are saying. Actually the cloning vector in the agrobacterium tumefaciens plants will be consisting of this mechanism. We have to explain in them about the RNAi mechanism, that means RNA interference phenomena, mRNA silencing phenomena we have to explain in the pest resistant plants. And apart from this thing, Bt cotton. Bt cotton is very very important, Bacillus thuringiensis particularly, and Bt cotton is always it acts against the bollworms. It is resistant against the bollworms. In the Bt cotton we have to write about the Cry1 AB and Cry2 AC genes, and how these Cry genes always they kill the bollworms. After going to the digestive system of the bollworms, what they are doing, they are releasing the crystals, and they are killing the particular, they are lysing by doing the lysis of the particular digestive system of the particular bollworms by releasing the crystals. How they are Bt cotton, that means Bacillus thuringiensis bacteria, they are killing the bollworms we have to explain here, but Cry genes we have to explain particularly in the Bacillus thuringiensis and all these things. And apart from this thing, in the 4 marks answer, biogas production. The biogas production, how much amount of the concrete tank we have to take, and floating chambers, the 2 inlets, about the waste material inlet and biogas outlet, that we have to explain about how the methane they are produced, and how the methanogenic bacteria which we had particularly, they are useful for the production of the biogas, how the biofertilizers are produced from the biogas. All these things we have to explain in the preparation, formation of the biogas and chemical nature of the biogas particularly. These all things are included in the 4 marks answers and short answers. Particularly in the short answers we have to remember whatever the points which we had particular, that points we have to cover in the short answers. From the genetics compulsory we are getting 1 question. From the first unit we are getting nearly 2 to 3 questions. Enzymes 1 question compulsory. Photosynthesis respiration 1 question compulsory. And plant growth hormones 1 question compulsory. And then actually in the molecular biology 1 question compulsory. Biotechnology 1 question. Last unit microbes and human welfare 1 question. All are easy, but we have tried the important points in the particularly very short answers. That means short answers particularly. These points you have to remember.